All right, well, welcome everybody. Thank you all for attending our first webinar of our new all-online biomanufacturing program this semester. I'd like to welcome Danny Grannick. Danny is a sales rep for Oxford Nanopore, which is a premier next generation sequencing company. I'm sure you've all seen their equipment and seen at least pictures of it or videos of it online. Oxford Nanopore has made a huge splash in the market a few years ago with the USB key sized sequencer that does end-to-end reading using a nanopore technology. And so Danny's going to be introducing us to that, how that technology works. He's also going to talk about some applications for industry as well as current research for COVID-19 and what's going on with the coronavirus. So Danny, welcome. And thank you again for delivering this content to us. We are very eager learners here, so go ahead and take it away.
Of course. Yeah, thanks, Doug. Can you guys hear me? Am I coming through okay?
Yes, you are.
Hear you good.
Awesome. Okay. You never know what problems are going to come up at the last second. So first of all, thanks for having me on. I'm really excited to be speaking to everybody today. I can't see any of your faces because I'm sharing the screens, but I want this to be a very interactive presentation. I'm planning to take a few breaks as I go through for questions, but I believe that we can submit questions through the chat room. There's also a method to raise your hand if there's something that comes up on a particular slide. I definitely don't want to be the only one talking for the entirety of the presentation, so please ask questions. I also kept this relatively high level, so if there's any more details or information that you want around a particular slide or even just questions that are completely unrelated to the content here, feel free to ask those. I'm happy to have a conversation around that. And then obviously any questions that you have after the presentation, Doug has my contact information. I'm always happy to chat with students and talk about where your interests are. So with that, I'll get started. The title of the presentation was kind of hard for me to land on, but I've decided on the new age of biotech. It's a very broad title, but I really do believe that we're kind of entering, or we're on the cusp of a revolution in healthcare and really the world around us, and it's being driven by a multitude of factors. But what I wanted to focus on is how we're using genomics and sequencing to really change the world around us. And this is everything from detecting, treating, and preventing disease to creating new materials and innovative industries. So I'm going to start with a very brief overview of sequencing and genomics, just to look at the space, and then I'll discuss Oxford Nanopore specifically and touch on our core technology. I'll go into a few of the key applications and industries that we're seeing genomics either really have a strong prevalence in or that's starting to enter. And then I'll finish up with an idea of what the future could look like using genomics and sequencing. So I wanted to start with the Human Genome Project. This is by no means the subject of the conversation, but I think it is a really good starting point. So the Human Genome Project, as I'm sure many of you know, was one of the initial attempts to sequence through the entire human genome. That's about 3.3 billion base pairs. And I believe it remains the world's largest collaborative biological project. The Human Genome Project, it coincided with the development of next generation sequencing. And I think, my perspective on that project is that it really catalyzed a lot of interest in genomics as an industry and the promise of that data. And I think it really accelerated development of a lot of the tools and platforms that we use today. So some quick statistics on the project. It took 13 years and it cost nearly $3 billion, and that was to produce a single human genome. They like to say it was complete, but in fact, in looking back at it, there were a number of gaps in the genome that we're still addressing today. But as I said, I think that the project really acted to accelerate the development of the field itself and a lot of the tools that we use. And I think it highlighted the value of sequencing as compared to more traditional genomic techniques. So as I'm sure a lot of you are aware of, genomics and next generation sequencing in particular doesn't require a hypothesis or knowledge about the targets that you're going after. So it enabled a lot of these scientists to sequence completely novel genes and identify new variants that, at that time, they thought would elucidate a lot of the mysteries around human health. I don't think that they had all of their questions answered with that single human genome, but as I said, it really spurred people to start looking into genomics as a really serious field to address human health. Next generation sequencing was massively parallel and scalable. We could, all of a sudden sequence through a lot more samples in a lot less time. And when you're looking at things like novel variants and new genes, being able to develop really comprehensive and large-scale data sets is going to be key for validating those findings and then translating those into actionable insights. And I think overall, next generation sequencing and the Human Genome Project, it was kind of this cornerstone, signifying the digital transformation in genomics, and we're still seeing that play out today. So a very brief overview of next generation sequencing. There's a lot of companies in this space, and they all use different methods. But on a very high level, what we can do is we can take your DNA or the DNA of several people or really any living organism, and we can extract it, and we can sequence that DNA, and we can deconvolute, the exact sequence of base pairs that make up an organism's genes. And what next generation sequencing really allows us to do is do that in a massively parallel scale. So now we can look at hundreds of samples or thousands of samples all at the same time, or we can sequence extremely large genomes like a redwood tree or the human genome at incredibly deep depth. And with the advent of next generation sequencing, we've seen a lot of companies pop up to address that market. So these are a few examples of the companies that make next generation sequencing platforms. They all use different chemistries and have different advantages over the other. So I think that we're seeing a lot of diversity in the market. And again, I think that's really contributing to acceleration and development of these tools. So one of the big takeaways is next generation sequencing has really enabled global scaling of genomic analysis. So we can sequence more samples for less money at a faster pace than ever before. And one of the kind of hallmarks that a lot of people in the industry like to use is Moore's law as an example. And you can see that the decrease in cost of sequencing has dramatically outpaced that of Moore's law, and it's only getting lower. So I think when we look back at the Human Genome Project and we use that 13-year, $3 billion statistic, we can today sequence a genome for less than $1,000. And I think that we'll continue to see that price drop. So with the decrease in price, with greater accessibility to those tools, we've seen widespread adoption of genomics in both traditional and novel industries. So, some traditional industries would be things like cancer research, microbiology, human genetics, clinical research, but we've seen this really interesting expansion in terms of the industries that we've been able to address. Anything from security for outbreak surveillance, which I think is obviously very relevant today, to more industrial applications around forensics and water testing and biopharma production, and even educational adoption where colleges and high schools want to internalize the technology to really teach their students hands-on approaches to genomics and bring genomics to the forefront of that educational journey. So that brings me to the company that I work with. So Oxford Nanopore, we're a relatively small early stage company, but our goal is to enable the analysis of any living thing by anyone, anywhere. It's a very lofty goal, but we're making pretty good headway, and our technology really centers around the use of a nanopore. So you can think of this as essentially a tiny hole. It's made out of a protein, and the way that our sequencing works is we have this nanopore embedded in a membrane. I don't know if you can see my mouse, but we will basically use a protein that will introduce DNA or RNA into that nanopore reader. And as the DNA or RNA is pumped through the nanopore, each base pair will emit a very significant and noticeable change in the electrical signal that's going across that hole. And you can see kind of a live demonstration of that electrical signal in the upper right-hand corner. So it's that squiggle. And what we can do is we can deconvolute that squiggle into the individual base calls for a particular sequence. It's an incredibly modular platform. We've made a lot of improvements over the last year, but the take-home message is that we've had significant progress on throughput, speed, and accuracy. All of the things that, again, are going to be drivers for the genomics industry. And I guess before I go on, are there any questions on the platform itself that I can address?
I'm not seeing any questions in the chat room right now.
Okay, cool. So I'm going to get a little philosophical right now. We've seen this incredible linear progression in the technology industry. So on the far left side of the presentation, you can see an old mainframe computer. These were incredibly expensive, centralized machines that required specialized engineers to run. And sorry, I'm seeing a few things in the chat. Okay. So these were incredibly centralized, specialized instruments that engineers had to run. So all of the computing was essentially being sent to a centralized, for lack of a better word, lab that was running all of your computational questions. And then we progressed to the advent of the personal computer and the laptop, and that transitioned those capabilities into the homes of individual people. So all of a sudden you had more people using this technology with direct access, and it propelled the industry forward in terms of the industries that it could serve. You know, there were a lot of new businesses that were formed around this transition. And again, we saw that transition with, you know, from the laptop to the iPhone, where you have this essentially a mobile computer that you can keep in your pocket and bring everywhere with you. And we were getting, again, more data sets, novel insights, new industries being created. And what we're seeing now is the advent of wearable technologies, you know, essentially a computer on your wrist. And I think with that, we're getting, again, more unique data sets, more unique insights, a lot of new businesses. And I think a really good example is biometric data. So with your Apple Watch, you can record the amount of steps you can take. You can record your heart rate. You know, there's new capabilities being integrated, but these data sets are providing really novel perspectives on the population that companies are using to create new industries or to inform lifestyle choices and things like that. So Oxford Nanopore, I think traditionally the sequencing industry has kind of followed this linear progression, and that a lot of companies right now have a very centralized, expensive system that requires an incredibly specialized faculty to run, and we're seeing centralization of the genomics industry. What Oxford Nanopore was really focused on was breaking that traditional cycle. So our first product launched was, was the MinION. So as Doug said, the MinION was, or is a portable sequencer. It's about the size of a small candy bar. And it lists for $1,000. So in increasing the portability and decreasing the cost of entering the field, we've made an effort to democratize sequencing. Again, getting our technology into the hands of more users that don't need to be specialized faculty to enter this world of genomics. And we've seen a lot of interesting data sets come out of that endeavor. In keeping up with the more traditional spectrum, we've released the Promethean on the left side of the screen. This is our highest throughput platform. You can imagine that you'd be running tens to hundreds of human genomes in a given amount of time. And then on the far right side, we have a kind of future product development roadmap to bring down the size of our sequencers even greater, to the point where you could plug it into your iPhone. So again, you can imagine that people can bring this technology into really novel environments. And I think we're getting some questions, so I'll take a second to address them. So the first one is, what are the practical benefits of having one's genome sequenced? So that's a really good question. I think it depends on the context that we're looking at. So there's things like cancer research where we're sequencing a patient's tumor to identify specific variants that may characterize their form of cancer, and that can inform optimal treatments for that patient. I'll dive into a few more specific applications. I think another really good example is the emergence of microbiome sequencing. So looking at gut bacteria and viruses and fungi, and then prescribing things like personalized probiotics to rebalance the microbiome. The second question is, are there any reasons people might need to sequence their genome more than once in their lifetime? Yes. So as you know, cancer is essentially a mutation of somebody's own cells and causes proliferation of those cells. Cancer isn't necessarily, we can predict, predisposition to certain kinds of cancer with a certain amount of confidence, but that certainly doesn't give us 100% confidence in our ability to mitigate against certain kinds of cancer. So I think as the genomics industry continues to develop, we'll see sequencing being used in more of a screening environment where we may be able to tell you with some amount of certainty what health risks you are predisposed to, but we'll also continually sequence you throughout your lifetime to try to do early detection of the disease. And then again, if you do come down with something, inform those treatment options. Let's see. So I mentioned that a pocket-sized sequencer, it opens up a lot of possibilities for novel data sets. So we were able to actually get one of our sequencers on the International Space Station. And I think that this is really just a testament to the idea that, these advances in technology, making things more portable and easier to use, can just open up experiments that were previously impossible with a centralized system. So the first industry that I wanted to focus on or touch on was biopharma. I think that there's, from what Doug told me, a lot of interest in this industry. Next generation sequencing has really enabled rapid progress across a wide range of industries. But I think it's had a particularly unique impact in biopharma in terms of personalized care and precision medicine. And I think that those are two really vague terms that are used pretty often. So I just wanted to take a second and define at least my interpretation of those terms. So when we talk about personalized care, to me, that's really using proactive healthcare rather than prescriptive healthcare. And what we can do is we could sequence a human being, and with enough data available, we could inform them of their susceptibility to disease later in life. So you can imagine that you could have your genome sequenced, and we would be able to tell you, with a certain degree of confidence that you're predisposed to heart disease. And what we could then do is make sure that throughout your life, you're taking steps to mitigate that risk. So, staying away from fatty foods, making sure that you're getting regular exercise. But I think that data can also be used with your healthcare providers to make sure that kind of on the regular checkup side, we're tracking your development and making sure that we're keeping an eye out for any symptoms that might be related to that susceptibility. Precision medicine, I really think to me, it's simple. It's getting the best, most effective medication to the person who needs it. So the current paradigm in medicine is that we essentially use a lot of broad spectrum treatments to fight disease. So you might use the same therapy for a whole group of patients that on the surface have the same disease, but on a deeper level are actually very unique in their genetic makeup and in their response to a certain therapy. So what we're seeing is that there's more personalized diagnostics using next generation sequencing that's also enabling us to prescribe much more personalized therapeutics. I think that there's three really interesting areas that next generation sequencing is touching on within biopharma. So research areas is the first. Sequencing and really genomics in particular have opened up new areas for research. I think the microbiome and cancer immunotherapy are two really interesting industries, and it's paved the way for the development of new medicines. Before next generation sequencing was available, we didn't really have comprehensive tools to assess a patient's microbiome and analyze the changes and develop therapeutics to address imbalances within that environment. And the same goes for cancer immunotherapy. Genomics and sequencing has just unlocked this entirely new perspective on how we can not only detect disease, but also how we can treat it. And then we see therapeutic development. So new areas of research like the microbiome and cancer immunotherapy allow for the development of more effective medicines, allowing for safer and more impactful treatments. And on the clinical trial space, we've actually seen a complete redesign of how clinical trials are run so that we can ensure that patients receive the best treatment possible. And what we've seen is in a lot of clinical trials now, we're doing genomic characterization of patients entering those trials, and we're using that data to inform, does drug X work better for a patient with this genetic profile versus another? And we can carry that through the clinical trial and make sure that that drug is being given to the patients that will respond the best versus another drug that might work better for other patients with the same disease. So here's a quick example. I won't go into the process of phage display, but I think that the take-home message here is that the scalability of next-generation sequencing is just allowing us to discover antibodies that we really couldn't detect before. So you can see that when you're doing a phage display, you have almost an incomprehensible number of potential antibodies that you might be screening for at any given time. And the scalability of NGS has just allowed us to find that needle in a haystack, allowing us to identify, the best antibody for a certain antigen or the best therapy for a certain disease and make sure that we're detecting those. With older technologies, I think a lot of those antibodies were getting lost in the noise. So we've seen kind of a growth in the diversity of therapies available as well. Before I dive into synthetic biology, I did want to take a chance to check the chat. Do we have any questions?
I don't think we have any new questions. Yeah.
Okay, cool. Synthetic biology is a really fascinating industry, and I think my colleague Ron had come by and talked about it, so I'll just do kind of a high-level description. But synthetic biology aims to harness the emergent properties of the central dogma for biotechnological and human use. And really what I think that means to me is we're taking control of biology and we're able to engineer it in order to produce the materials or the things that we want. It's a kind of the perfect definition of biotechnology in my mind. So synthetic biology has an incredible amount of industries that have kind of spurred out of this general term. There's companies that are using synthetic biology to make new medicines in pharma. There's companies that are using it to optimize agriculture. There's companies that are using it to replace the chemical manufacturing process as well as the materials manufacturing process. I believe Join Bio, Pivot Bio, Clara Foods, Lygos, Zymergen, Bolt Threads are all in the Bay Area. So, I think as students and even myself included, I think we're all really lucky to be geographically located in this hub for innovation. We're seeing a lot of the early adopters of these techniques come out. So the synthetic biology process flow, I think that there's a lot of interpretations of what it looks like, but I like to think of it as a 5-step process for discover, design, build, test, and scale. And there's going to be constant iteration between your design, build, and test phases for any synthetic biology workflow. But sequencing really applies to every step of this process, which is why I think synthetic biology as an industry is really fascinating to look at from both a genomics and a sequencing perspective. So the discover phase, that's really the identification of a source compound and its corresponding gene sequence. Bolt Threads is just an awesome example. This is a company, I believe they're based out of East Bay, and they were able to identify the genes involved in creating spider silk in spiders, and they've been able to transfect those genes and optimize them, and they're able to produce those at scale using synthetic biology methods. So we're basically taking the spider out of the equation and making spider silk, and Bolt Threads is weaving the spider silk into clothing. And there's some really interesting properties of spider silk that lend it to be incredibly useful in clothes. So spider silk is antimicrobial, it's incredibly strong. So you can imagine that, if we were able to really scale up production of a material like spider silk, there would be huge benefits in terms of the clothing that we wear. But it all starts with the determination of the source gene sequences, and the determination of those sequences is obviously based in next generation sequencing. So it'll be in gene identification. They'll often look at different species or different strains of a certain bacteria to make sure that they're optimizing production of that material and identifying any novel variants that may influence that production. So there may be a certain variant in a certain kind of spider that produces, a slightly different version of spider silk that for whatever reason is better for them to use in their clothing. And that's just an example of kind of what that discovery phase looks like. And then you move on to design. So a lot of that data is loaded into data repositories and annotated, and then it's either shared or maybe kept private within these companies and researchers. But there's a lot of public tools available that actually allow you to access this sequence data and gene circuit data so that you can understand kind of the basics of how you could create something like this. The next phase is build. So for this, I just wanted to highlight the fact that almost every company sees DNA sequencing as being essential to their business. You'll notice that some companies prefer to have that in-house versus outsourcing to something like a commercial service provider. I think the study is a few years old, and I'd be interested to look at the numbers today. I do expect that to shift for the same reasons that I discussed earlier, just lowering the barrier to entry to sequencing. I think we'll see a lot more adoption of sequencing technologies into synthetic biology companies themselves, just to have more control and faster turnaround time on getting the data that they need. So once you have identified your genes, you need to synthesize them. So there's a couple companies that do this. IDT is, again, located in the Bay Area. So there's a lot of quality control that's required to make sure that the genes you're synthesizing are the ones that you want. And what a lot of companies will do is they'll produce degenerate versions of a gene so that they can test several variations. So there's a lot of value in being able to look at those degenerate sequences and make sure that you know exactly what variants lie in them. And then you'll want to test it. So once you have your gene circuit or a certain gene of interest, you'll transfect it into something like bacteria or yeast. You'll perform mutagenesis so that you get those degenerate versions of that gene. And then you'll perform some kind of experiment to determine what your high producers are, how much they're producing and what those genes look like within those organisms so that you can then scale up production from there. So there's a little piece in here. Our ability to create DNA de novo is not equally matched by a capacity to predict the ideal DNA sequence a priori for a given application. So basically what that means is while we're able to some extent predict an ideal gene sequence in silico, so using software, it hasn't gotten to the point where it replaces a wet lab experiment. So that sequence information in next generation sequencing platforms are still going to be critical in being able to deconvolute the profile of a successful gene and what that looks like versus an unsuccessful gene. So for each step, there's been respective companies that have popped out. So in the design space, we have companies like Benchling, and they're based in San Francisco. For editing, there's a really interesting company called Inscripta. I believe they're located in Colorado. For gene synthesis, there's Twist, again, here in San Francisco, and an earlier stage company called Molecular Assemblies based in San Diego. And then for the actual engineering piece, there's companies like Zymergen, again, in East Bay, and Amyris, which I believe is also in East Bay. And I think to my earlier point around kind of being at a hub of innovation, East Bay is a particularly good spot for synthetic biology. So there should be a lot of opportunities to get involved over there. Okay, before I dive into public health, I just want to check any questions. Do we have anything in the chat?
Not seeing anything.
Actually, I just saw something from Rachel.
Zymergen is around the corner
okay.
from my house.
Okay.
Yes. So they've probably grown quite a bit in the last couple of years, depending on how long you've lived there. They occupy a few buildings now, but they're definitely a leader in the synthetic biology space, and they're growing even more now. So with the prevalence of SARS-CoV-2 and COVID-19, I wanted to take a second to talk about how sequencing is being used in the public health space. And I think sequencing and genomics really applies to everything that you see on that poster, but I'll do a deeper dive. So there's kind of three phases to addressing a pandemic. The first being an acute response. So an acute response is what we would want to see within the first 1 to 3 months of a pandemic. And sequencing in this case is really being used to analyze and trace the spread of SARS-CoV-2 globally. What this will do is help track and contain the virus within populations. So this is purely a screening application, making sure that we understand exactly what kind of strains are out there and where they're occurring. Following that, we have precision isolation and exit, and that usually encompasses the next 3 to 6 months. But we need methods to identify and isolate the at-risk population in order to minimize severe cases and mitigate overload in our care system. So NGS is being used to develop profiles that will characterize a patient's susceptibility by looking at genomics within the host. And I'll get more into detail on that in a few slides. And then last, and this is kind of a continuation of everything. So it'll be, after about 6 months, but usually it'll be a permanent solution in place. But we need to think about, how do we develop a testing and analysis infrastructure that will properly address events like this in the future? And how do we conduct ongoing surveillance of the population to minimize recurrence? And I think that the ongoing surveillance piece is really interesting. So with the democratization of sequencing, what we're discussing with organizations like the CDC and the FDA is our ability to basically deploy these small, portable, inexpensive sequencers so that when something does pop up, we have the tools needed to address it right away. And we've seen this kind of adoption across a few other industries. So we've worked with the CDC and the FDA on tracking outbreaks in food. So you can imagine that the CDC and the FDA would have a lot of interest in being able to address something like an E. coli outbreak from a food production facility and being able to trace that as quickly as possible and understand where it originated. So this is an example of what some of the data would end up looking like. There's a organization called GIS Aid, and they have a great website. You can look at all of these tools live and interact with them, but this is just an example of what the genome of SARS-CoV-2 looks like, and then these are a couple of use cases of how that data can be displayed, but you can see, exactly how many strains there are and what countries those strains are attributable to in the bottom left corner. And then in the bottom right, you can actually play a live video of the spread of COVID-19 on a global scale. And a lot of that information is coming from sequencing specifically. The Arctic network is a network that we have partnered with to develop a specific workflow around sequencing coronavirus. So when you're developing, a workflow to sequence a virus that's part of a global pandemic, it's really important that you have a really fast turnaround time, that you can sequence a lot of samples at the same time, and that you can do it for a relatively low cost. So we've been able to do that with Arctic. So we're able to sequence viral genomes in about an hour on our technology at about $50 a genome, which has just enabled adoption of that technology into the public health space and allows people to actually use the technology to analyze the virus. So in parallel to our work around sequencing the virus itself, there's a lot of interest in looking at the patients that contract the virus. And there's two really large areas of interest around co-infection and then host immune response. And you can use next generation sequencing technology to look at these as well, oftentimes in the same assay. So co-infection is simultaneous infection of the host by multiple pathogens. You can imagine that somebody could come to the hospital presenting with coronavirus as well as a bacterial infection. And what next generation sequencing can do is detect both of those pathogens. If you have a patient that comes in with multiple pathogens, including coronavirus, but coronavirus is at a relatively low titer, which means that they haven't been badly exposed to the virus, it may be that a lot of their symptoms are actually connected to the bacterial infection, but their immune system is compromised as a result of fighting coronavirus. So you can make sure that they're getting the right treatment to address the pathogen that's causing the most damage. But you're also mitigating the idea that they are a coronavirus patient at the same time. For host immune response, so there's two versions of the immune response that we focus on. The first is the innate immune response. So that's the first line of defense against pathogens, and that occurs within the first 0 to 96 hours of infection. So this presents itself with symptoms like a fever or swelling, but it's a very non-specific response to prevent the spread of pathogens throughout the body. So something that we've been able to do, and we've partnered with a few researchers at UCSF on this, is to conduct RNA sequencing on patients. And what you can do is you can look at the gene expression profiles and develop a signature where if a patient comes into the hospital, you could perform RNA sequencing, and that gene expression profile can inform you as to whether their innate immune response is in response to a viral infection, a bacterial infection, or if it's an autoimmune disease, which again can inform treatment. The second kind of immune response is adaptive. So that's an acquired or specific immunity. It's incredibly specific to the pathogen that's presented, which means that your adaptive immune response would be in response to coronavirus itself, not just a bacterial or a viral infection. It's a long-lasting response over 96 hours, and the key mediators are T and B cells, B cells being the ones that ultimately produce antibodies to fight off the foreign invader. And what our technology can do is, again, sequence T and B cells in patients that have coronavirus, and that can inform us on how that adaptive immune response is working. So you can imagine that somebody who isn't responding well to the disease may not be producing antibodies, or may not be producing antibodies that are working well enough to destroy coronavirus, but we could also sequence the B cells and T cells of a patient who has a very quick and rapid response to the disease, and that can guide us in terms of how we design antibody-based therapeutics downstream. So the platform on the left is one of our sequencing platforms, but the take-home message here is that you can essentially get information on the virus itself, on co-infections on the patient's own genomic background, all on the same instrument and oftentimes with the same assay. So you're able to get these really diverse perspectives at scale. That's just accelerating our ability to make an impact and really fight this. So all of these applications apply to the chart on the left, but I think moving forward, we'll see continuous patient and population screening, and that involves both viral and immune response characterization. NGS can inform viral evolution, providing insight into the efficacy of vaccines and therapies, as well as new potential targets. So, as we're sequencing the genomes of these viral strains, it's important to note the variations between different strains, because it's possible that the vaccine you create for one strain is not going to be effective in training the body to fight off another strain. And that's kind of what we see with flu vaccines every year, where we're really only fighting off a subset of the possible strains of the flu. But there are some strains that our bodies aren't ready for. And finally, NGS can guide treatment options for patients and inform co-infection. So that's it for public health. And I wanted to check one last time on any questions.
I have a question, Danny.
Yeah.
We have like local companies in the Bay Area, like SynBio Bio and BioIndy. I know that they're holding like COVID town halls every couple of weeks. And it seems like they're collaborating at a faster pace with other biotech companies just to come up with solutions for COVID. Has that changed how you work with other companies as well?
Yeah, that's a really good question. I think organizations like SynBio Beta are really good at bringing a lot of diverse perspectives to the same table to engage in those conversations. And they've done a really good job of catalyzing a lot of collaborations between researchers as well. I think when you're faced with a global pandemic like this, the actions that you can take to address it often outweigh kind of more typical business concerns. So I think it's changed the way in my company works, in that whereas, for some of the assays that we're rolling out, we typically wouldn't engage in collaborations, but in a situation like this, really anything that you can do to accelerate a solution, is kind of a necessary step to take. So I think that those kind of town hall meetings have just been great to get those conversations started. Does that answer the question?
Yeah, thank you. I have one more question to you. The Promethean versus the MinION, can the MinION do the multiple assays too, or is that just with the Promethean?
Yeah, so the MinION can, it's less about multiple assays and more about the data that you can extract from a single assay. So what we can do is there's a really interesting methodology. You can see it under metagenomic sequencing. It's called MSSPE. And what MSSPE is basically a method to do a low-pass shotgun metagenomic sequencing. So we're really just doing a bulk RNA assay. So we're looking at the host transcript as well as the pathogen transcripts. And what we can do is we can actually spike in targeted primers for that assay to focus on certain pathogens of interest. So you can imagine in an RNA sample, you have a whole lot of host transcriptome. You may have a whole lot of a bacterial infection, and then your SARS-CoV-2 may be minuscule, and it could be something that you miss in a lot of assays. So by spiking in those primer sets, we can enrich the signal of that SARS-CoV-2 virus. So you're actually getting the full genome of the virus. You're getting the co-infection information. And in a lot of cases, you're even getting that gene expression profile of the patient to get innate immune response. But to my earlier point, for the MinION, we have barcodes available, so you can actually barcode multiple samples together and run those. And then the same goes for the Promethean. It's just at a much larger scale.
Okay, thank you.
I thought I saw something come on the chat.
Yeah, there's one more in the
so
chat.
How is the rate? Yeah, how is the rate of NGS affected by the smaller form factors, such as the MinION compared to benchtop sequencers, if at all? So that's a good question. I think it depends on what your definition around a benchtop sequencer is. So it's more of a, I think it's a great chemistry question, actually. So something like Illumina that uses sequencing by synthesis, where they're essentially rebuilding fragments and taking images of those individual nucleotides as they're incorporated on the strand. It's a very accurate process, but it's also comparatively slow as compared to a nanopore sequencing method. So in the slide that I had earlier for nanopore sequencers, each fragment is going through a nanopore at a rate of 450 base pairs per second. You can imagine that on something like a MinION, we have 1,500 of those nanopores all sequencing in parallel. So you can imagine that your rate of data production is incredibly fast as compared to an Illumina sequencer. And you're actually getting those base calls in real time, so we can perform analysis while we're sequencing. So I think that the rate of NGS is more dependent on the chemistry and then the scalability of that chemistry rather than the size of the sequencer itself. Okay. So we might have one more question. Oh, never mind. Okay. So the future of NGS. Let's make sure this works. So the intersection of technology and biology through insights obtained from big data, and big data in this case is really the emergence of next generation sequencing has fueled the drive toward precision medicine and personalized health initiatives. So we're seeing big data in biology. It's producing a lot of progress in precision medicine, which enables patients to receive highly specific and effective treatments based on their background in specific disease. And it's opening up the door to personalized health that can inform susceptibility to disease for lifestyle management and proactive rather than prescriptive care. And those two kind of factors are coming together to really form what I would consider a new standard of care. And I think, this is still, it's a roadmap that's being worked on. And I think that we're kind of at a phase now where we are producing a lot of genomic data, and the next phase is going to be translating that genomic data into actionable insights and then rolling those out to patients and really the greater population. So that's things from kind of the health perspective. And then I did want to take a slide and talk about some new applied markets that are being opened up. Some of these are being opened up just from next generation sequencing and genomics in general, and some of them are specific to Nanopore and our decentralization goal. The first is eDNA, or environmental DNA sequencing. And I touched on this earlier, but it's really a question of, how can we use genomics and sequencing to enable for continuous monitoring of our food and surroundings for potential pathogens? And I think that the idea, we're not too far off from the idea of having a sequencer in your house that you're using to not only test the environmental DNA around you, but that you might actually use for your personal genomics, and then that data gets uploaded into a central repository and it gives you some kind of report. So to the earlier question, one day you may end up you may be sequencing yourself at home as part of a screening procedure based on your original whole genome sequence that gave you a risk profile of diseases that you might be susceptible to. The second applied market is DNA data storage. So there's been a lot of advancement in this industry, and we're seeing a lot of partnerships open up around using DNA as a medium for storing data. So DNA is incredibly dense. It's orders of magnitude denser for storing data than current methods. Incredibly easy to replicate. A simple PCR reaction can give you billions of copies of the same information. And it lasts for a long time. So we've sequenced samples that are thousands of years old, and there's a lot of interest in using DNA as a storage medium for a lot of archival data sets. So these are older kind of bank records and health records that we need to have on hand, but probably won't be accessed for a long time. And we can really use DNA to replace these huge server farms. And sequencing is obviously the reading part of that, like a computer would be used to read data off of a CD or a USB drive. So I think we'll continue to see emergence of that kind of technology in the next couple of years. And the last one was just personalized products in general. I actually saw my first microbiome-based skincare commercial a couple months ago, and I was shocked by it, but I was also really impressed. But we can leverage our genetic makeup to receive incredibly personalized consumer products, including, deodorant, skincare, and even probiotics. It would be a little bit dystopian, but I wouldn't be surprised one day to see targeted ads, not only based on your browser history and what you're searching for, but also your genetic background. So, at the end of the day, I think genomics is progressing the health industry by leaps and bounds, but I think it's also catalyzing a lot of these really novel, innovative industries to start from the ground up. And we're only just starting to see the emergence of these companies and industries. But, I think our hope is that in the next decade, a lot of these are commonplace products, and they really, serve to help humanity. But that's everything that I had for this presentation. I hope you enjoyed it and got something away from it. So I wanted to open it up for any questions, but like I said, I'm always happy to chat or follow up on, any particular interest or do a deep dive on anything.
Danny, thank you so much. That was a great presentation, super informative. So I did have a question. And I was curious, in your interaction with your customers, and you do a lot of academic labs, but you also probably have clinical labs as well. Are there people running their own, like say you're an academic lab, you want to know if you have COVID or have had it, perhaps. Do you know about people that are using your product or anybody else's that doing kind of self-testing, to
Yeah, that's a great question. Ironically enough, we had an internal phone call last week, and our company is actually doing internal testing of any employees that think they have or have been diagnosed with coronavirus, and we're using our platforms for those kinds of point of care settings. We're still a few developments away from having a truly kind of point of care platform. So you still have to do upfront library preparation, which is really, a few chemistry steps to prepare your genomic material for compatibility with the sequencer. And then there's some downstream advancements that have to be made in terms of interpretation of the data. So I don't think we're at the point where anybody is, performing a sequencing experiment in their kitchen, although it is very possible. I would say, anybody who has lab experience and has a MinION could absolutely do that in their kitchen. I'm not endorsing it, but it's definitely possible. And I think that we'll continue to see that popping up. There's academic labs that are investigating those types of applications, and it's a matter of process validation, but I think that we'll see the emergence of case studies like that in the next couple of years.
Thank you.
Yeah, of course.
Hey, Danny. I have a specific question for the MinION that you mentioned earlier at the beginning. You said that it's the size of like a candy bar. So then, like, how they gonna clean it like after use, and like how often they have to like calibrate it like that?
Yeah, that's a really good question. So I think, part of our goal around democratizing sequencing is not only making a smaller platform, but making a simpler platform. So the MinION doesn't require any kind of cleaning steps. All of the chemistry is in a flow cell. It's a separate component that you put into the MinION. And let me see if I can just pull that up. So if you look at the MinION, you can see, and I don't know if you can see my cursor, but you can see kind of the metal casing that's the sequencing platform, and then inside the metal casing is the actual flow cell, and that's where the sequencing kind of the magic sauce is, for lack of a better term. So that flow cell is a single time use, and it just clicks in. So the sequencer itself is incredibly robust. We've had people bring it on ships out to sea to do live marine sequencing. We've had people take it on airplanes. I got a video sent to me the other day of somebody who strapped it to their bike, and they were sequencing while they were biking home. So it's an incredibly robust platform, and we've tried to make it as simple as possible to use and take out a lot of the steps that come with a more complicated technology, like washes, like recalibrations. It's really something that's not required with our technology.
Thank you.
I have a follow-up question on that. So I was looking on your website, it says like it's $1,000 for the basic of the MinION. Like how much do the flow cells cost and other reagents and stuff that go into it as well?
Yeah, so the MinION itself is $1,000, and with that, we'll send you 2 flow cells and a library preparation kit. And the goal there is to really just lower the barrier to entry for anybody to get started with our technology. After that, the flow cells are, for the MinION, are for single flow cells, $900 each, but as you increase your volume, that price drops dramatically. So I think for a pack of 48, the price drops to $500 per flow cell. And with that, we've also released lower throughput flow cells. So each MinION flow cell will get anywhere from 15 to 25 gigabases per run. But we also have a flow cell called the Flongle, and that flow cell will get about 1 to 1.2 gigabases per run, and that's a single-use flow cell, totally disposable, and that's $95 each.
Thank you.
Yeah.
Well, we could take a few more questions if there are any out there. I just wanted to say I put in the chat the info for our next talk that we're going to have as part of our ongoing effort to bring in experts to our biomanufacturing program to talk on different topics. So if you look at the chat, and some of you may be calling in, you may not be able to, just email me if you haven't gotten this info. My email is dbruce@peralta.edu. You can email me that way. But yeah, so the next talk will be on downstream processing. So maybe not as sexy as DNA, but really cool topic, and I hope many of you can attend. We have someone from the Advanced Biofield Process Demonstration Unit coming to the webinar to talk. His name is Jean-Philippe Prahl, and he'll be talking about how to do the downstream part of purifying protein, analyzing it, things like that for contract manufacturing, which is what they do. So that's on May 5th, a week from today, at the same time. So we also have a YouTube channel that I started up. I put that in the chat as well. I'll post talks like this in our YouTube channel so that we can kind of create an online presence, and you can check that out in the chat. So I did have one other question, Danny, too. So you mentioned how you the shift toward in-house versus outsourcing sequencing. We only recently started using an outside sequencing service in the Bay Area that's local to us, and I'm just surprised how cheap it is. It's and they even pick up your samples and then for free, and I just don't know how they're making any money at all. So it seems like it's just a razor thin margin to begin with. And it's probably just one push away from, like you said, in-house adoption, these technologies you're talking about. But can you maybe contrast what the Nanopore does briefly with other sequencing? There was a question about benchtop sequencers. Now, long read, as I understand it, you may lose some fidelity or accuracy, right? Whereas you might need other traditional, maybe even Sanger type methods to get accuracy on certain applications. Isn't that correct?
Yeah, so right now Nanopore is a little bit less accurate on a raw read standpoint. So that's an individual fragment going through a Nanopore. What most applications will do is they'll get a consensus sequence for a given sample, where they'll basically stack reads together and extract a sequence from there. And For that route, we actually have much higher accuracy than a lot of other technologies, and we're working to improve our single molecule accuracy. To your point, though, I mean, I think that there's certain applications in certain situations where it still makes sense to outsource your sequencing. We're not at the point where internalization is an apples to apples comparison. So for certain companies, you know, like I said earlier, there could be companies that require huge amounts of data coming off of their sequencers. And for a lot of technologies that produce that amount of data, you know, the platforms can be $350,000 to $1 million, and that's just to get the platform for your lab. So while you know, Oxford Nanopore has a slightly different model that allows people to bring in those high throughput platforms on their own. You know, a lot of companies aren't able to internalize a platform like the NovaSeq that costs $1 million, and it requires them to outsource. But I think one of the sacrifices that you make is the turnaround time and getting your data back. And, you know, I think in a lot of my conversations with the synthetic biology industry, you know, when you have experiments where you have to iterate a lot, the turnaround time between getting data back on those samples, you know, can take weeks, and that really impacts your ability to make decisions and move forward with certain kinds of genes or certain, you know, bacteria that you want to implement into your processes. So I think it's a delicate decision for any lab or any company to make. You know, I think our hope is that we want to make it not a decision, you know, to the point where from a financial standpoint, along with all of the other points, it just makes sense to internalize your own technology. But for now, access to some of those high throughput applications just has a barrier to entry that's relatively inaccessible, especially if it's a small startup company or something like that.
Great, thank you. Are there any other questions for Danny? Okay, well, it looks like there aren't any more questions, Danny. Thank you again for this wonderful presentation. And it was very exciting to see how the technology is progressing. I hope to see that new iPhone version somewhere soon. That sounds pretty cool. And yeah, once everything's back to normal, hopefully we can have you in our lab someday to check out our lab, and maybe we do some product demo or something like that too.
Yeah, that'd be great. Always happy to come by. And everybody, thanks again for having me on.
Great, thanks. All right, everyone. We'll see you at the next talk, all right?
Mm-hmm.
Thank you.
Thank you, Danny.
All right, thanks everybody. Bye.
Bye.